Alright, so today we're learning about topic 1.6, which is cell division. Uh, as part of cell division, there is the cell cycle, and there are three main phases of it. There's interphase, mitosis, and cytokinesis. And interphase is the longest, as you can see in this image here. It's this big part right here, because it contains G1S and G2 phase. G2, or G1, is essentially just the normal cell growth of the cell. And then you have S phase, which is the DNA replication in the nucleus, and this is to prepare for mitosis. Then you have G2 phase, which is the protein synthesis, as well as just preparation for the cell division, and the next phase, which is mitosis. Now, mitosis is the actual division of the nucleus into two daughter cells. As you can see in this image, there are four main phases. There's prophase, and then there's metaphase, where the sister chromatids form. This is where all the chromosomes lay. There's anaphase. This is where the spindle fibers begin to pull apart the sister chromatids on the centromere. And then you have telophase. This is when the nuclear membranes begin to form. And next you would have cytokinesis. This is the last part, and this is the division of the cytoplasm into two daughter cells. And it's different in animal and plant cells. In animal cells, a ring is produced of microfilaments to form a cleavage furrow, and then it pinches off into two daughter cells, and there's your division. In plant cells, it's a little different, because they form vesicles to form a new cell wall, and they split into two daughter cells. So basically, the main difference is plant cells create a cell wall, which is very obvious. Now we have cyclins, which are very important. They control the progression of the cell cycle, as you can tell in here. They bind to CDKs that attach to phosphate groups to trigger the phases. Cyclin D occurs G1 phase, and then it carries through the whole cycle. Cyclin E is at the peak of S. Cyclin A sets off G2 phase, and cyclin B sets off mitosis, and it aligns the chromatids and spindles, as we saw earlier. And we also have tumors, and the formation of a tumor is just from mass amount of duplication of cells, and this occurs when a cyclin is disturbed and mutated, and then the cell is no longer able to enter G1 phase, and you can tell that someone has cancer cells or a tumor, because the cell is, for the most part, in mitosis, because it can't be in G1 phase. It just keeps duplicating. We have two different tumors. Benign tumor, I'm pretty sure, what it's called, and that's just a localized. It's easier treated. And you have the malignant, which is more cancerous, and harder to treat. That's the one that spreads through your body. And that is all of 1.6. Now please like and subscribe, or else, I don't know.